Welcome to Marie's Lab. Now, before I start, if this is your first time visiting this channel, kindly like, comment, subscribe and share. If you've always been here with me, kudos to you. In today's episode, we'll be talking about the Microbiology department. Now, the Greek word micros means small and bios means life, then logia means study of. So basically, microbiology is the study of microorganisms. And what are microorganisms? Microorganism is any organism that are too small to be seen with the eye, except with the use of a microscope. And what are some of these microorganisms? Some of these microorganisms are protozoa, bacteria, algae, fungi, yeast, and viruses. And these microorganisms can be grouped as unicellular, that is single-celled organisms, multicellular, that is two or more organisms, and acellular, that is organisms that lack cell or organisms without cell. Now, in this very department, we normally use aseptic technique. Let me bring it down to the layman understanding. So aseptic technique here means the measures we put in place during the sample taking and during sample processing in order to avoid contamination of the sample. That is aseptic technique here. And in this very department, we normally identify organisms by culturing your sample. So we normally identify the organism by culturing and doing sensitivity. And sensitivity here means being able to find the best antibiotic for the organism we've identified. So let me chip in this. Whenever you visit the laboratory and the laboratorian informs you that your test is culture and sensitivity, please don't be in a rush for your results, because your results may take 3 to 1 week for it to be ready. Now let's look at some of the samples that are being run in the microbiology department. We have urine routine examination, stool routine examination, high vaginal swab routine examination, stool culture, high vaginal swab culture, acid-fast bacilli for sputum, Monto test, skin snip, skin scraping, wound swab culture, and the likes. Now let's look at some of the media we use to grow these organisms, or some of the media that we use to grow or culture these organisms. We have the cysteine lactose electrolyte deficient, blood agar, chocolate agar, thiamartin agar, bile esculin agar, mannitol salt agar, sulfate citrate bile salt sucrose agar, and the likes. I believe this has been very helpful and you would like the next person to benefit from this. Kindly like, comment, subscribe and share as you make an impact. See you in my next video. Bye bye.